Four times 5-minute visits. So you're going to visit 4 laboratories, 4 different laboratories, for 5 minutes, 5 to 10 minutes each, and then we're going to swap. Just a few words about this building. So as I mentioned in the lecture, it was designed by Norman Foster. The idea is that these are 2 different hemispheres of the brain. So this is the south hemisphere, this is the north hemisphere of the brain. And these stairs that connect the left and the right sides of the brain are like the corpus callosum, which are the fibers that connect the left side of the brain to the right side of the brain. The building is designed in such a way that it will promote interaction between the different disciplines, as I mentioned. One way is these common floors. We call them the social hubs. So this is a place for students and faculty to interact among themselves. But the brain is also connected within the labs. You will see glass doors that people can walk, students and faculty can walk freely between the different labs inside the door. Inside the building. The building itself is 5 stories high. -2 is the parking lot and computer center. -1 under the courtyard is a big animal facility, plus all kinds of other facilities that are shared facilities that are served by all the faculty of the center, but also many researchers around the universities. This is the courtyard, which houses the administration, the classes, including the auditorium. If you use your imagination, on the left-hand side there will be hopefully a cafeteria next year. We were preparing, but with the corona pandemic, this thing stopped. And then the upper 3 floors are the floors of the laboratories. As I mentioned, we are a very interdisciplinary bunch of researchers with very different disciplines. So now we're going to split. I'm going to take the green group. Everyone with a green bracelet is going to go up another floor. Orit is our executive manager. She has the red bracelet. Everyone with the red bracelet can follow Orit too, and we meet on the social hub of the 2nd floor. 2 other groups go with Adi Sheri, the yellow group, and Alona with the blue, and we're going to split. They're going to take you to different labs, and we're going to swap, and it will take us about 35-40 minutes, so please don't delay. So green and red, we go one.
Blue group here. Yellow group, come with me.
Yellow?
Here.
we're going to go into one lab. Actually, it's my lab. Then we're going to go to Nomi Habib's lab on the other side. And then we're going to go down to two other labs and then go finish this.
Welcome to our lab. My name is Libby. I'm a PhD student of AD, and I'll tell you a little bit about what we're doing here. So in the lab we focus on studying plasticity, which is essentially the changes that our brain undergoes while we experience things, while we learn. Our brain isn't a static organ or a static machine. It changes through our entire life. So we focus here on the lab on plasticity in three main topics. So the first one is plasticity in the field of neurogenesis. I'm not sure if you know this fact or if you're aware of the fact that we are all born with some amount of neurons, or brain cells, and unlike other tissues in the body, this is the same amount of brain cells we're going to have through our entire life. We're not going to be able to manufacture new neurons, unlike skin cells or other tissue cells. So this is true in all the brain, except for two specific niches in the brain, which is the olfactory bulb, which is like the first processing station of our sense of smell, and a specific region in the brain which is called the hippocampus, which is in charge of memory formation. So we study here adult neurogenesis in the olfactory bulb. I'll show you an example if you can shoot here. Sorry, it's not on a bigger screen, but we are studying. I'll try to explain what that means that we study adult neurogenesis. This is a mouse brain, and now you will see we have a specific genetic system that allows us to target and mark these adult-born neurons. And you see all these light, bright dots are adult-born neurons. They are formed here in a specific niche in the brain called the SPZ, and we can track them and look at them after they are born and arrive to the olfactory bulb. And this is a 3D video of an olfactory bulb of a mouse with all these bright dots are genetically tagged adult-born neurons. And after we tag them, we can track them and see how they influence neural processing. What do they contribute to processing? Why are they, try to understand why are they so important and why are they preserved in these specific niches in the brain. Hopefully in the future, this type of basic science will enable us to harness this information and maybe use this knowledge to help with some neurodegenerative diseases or places where we would like to harness the power of adult-born neurons to help with some pathological conditions. So this is one example for plasticity. Other examples of plasticity that we study is learning. So this is a pretty intuitive example, because while we learn, the connections between neurons in the brain change it, and this leaves a mark in our brain. So we use here different learning paradigms. For example, my PhD is about category learning, how specific categories are grouped and encoded together in the brain. And so we try to see, to track these differences in the brain before and after learning to see what changes. And the third example of plasticity that we study is maternal plasticity. So, well, essentially animals, and especially mammals, female mammals, when they become parents, there's many hormonal changes that influence the brain. And this induces some maternal behaviors which are, they are not studied in an explicit way, but there are changes that happen inside the mother's brain that make her react to pup vocalizations, retrieve pups, take care of her infants in a way that naive females or males do not behave. So we try to track these changes, see where they occur in the brain, what type of behaviors they facilitate, etc. So these are our main topics of research. And the special thing about the lab is that we use a lot of very advanced and like a wide array of experimental techniques, from genetic systems to imaging, which means microscopy. This is actually this specialty and the lab specialty, and in a moment we'll show you an example of one of the microscopes that we use. The third main technique that we use is electrophysiology. So, as you probably know, neurons in the brain interact with each other using electrical signals. And if we measure electrical activity in the brain, we can know which cells were active, which regions were active, and try to decode what type of communication our neurons in different brain regions have. So this is the general scheme of what we do here in the lab. And now, if you will all be kind enough to follow Henri, he'll show you the microscope he's working on. It's a 2-photon microscope. He'll explain all of you what that means, and this is here in this room to your right. And after that, if we'll have a few minutes, and some of you, if you'll be interested, we can show you an experiment going on live right now with electrophysiology, but this is only if we'll have time.
So that's on the left room, but only if you want. There's a mouse that we're recording from the brain there, but maybe half of the team can walk in and Henri can give you a 2-minute explanation. Yeah, you can go in. Perhaps not everyone. And then we are going to go to another lab.
Hi. Nice to meet you too. And so here, this is where the magic happens. You can see all this system is meant to engage with the mouse while we measure neuronal activity. So I train the mice on tests, on perceptual tests, which improve their behavior, their engagement with the test. They learn how to engage with my system in order to get reward. So you can see here I have many things directed at the mouse at the same time. I have a speaker through which I can display sounds to the mouse. I can display odors to the mouse through this system that is connected to a bunch of jars that have odor liquids inside them. So I can deliver odors to this. I can deliver sugar water reward to this part. I take a picture of the mouse while he's engaged with the test, and I shine infrared light on it so I can see the picture in the dark. And the most important part is this. So this is connected to the entire system behind. This is the microscope. This is a super strong microscope that can go deep into the brain tissue and look at different neurons within the brain and how their activity looks like.
With all the scallop, so you go beyond the scallop.
So maybe I'll find a picture here. So the thing I do, I implant a glass window right above the auditory cortex of the mouse. And you can see all the blood vessels. This is the mouse brain. So I make this very small round piece of the skull transparent. I replace it with glass, and through which we can look deep into the brain. And this is how the data looks like when we look into the brain. So these are neurons in the auditory cortex, in the brain region that codes for sound. And you can see them glowing in green every once in a while. The green fluorescence represents the activity of the neuron. So we genetically modify the neurons in the mouse brain to glow green whenever they're active. And then we can track the same neurons over days and weeks and months and see how the way they respond to external stimuli changes as a result of learning. So this is how it looks like. Here I have an example of a mouse engaged in a test. So here you can see this is this camera, this is a video. So you can see the mouse every once in a while, it clicks the port when it knows that it should. So this is a mouse that learned when licking is appropriate. You see it wanted to lick now, but it stopped itself. So we can't hear the stimuli here, but whenever the correct sound, whenever the mouse hears the correct sound, it clicks. So I built this microscope, and additionally I wrote a program with many parameters that I can modify.
What do we need to solve?
That's it. Alright, so this is the mouse brain, this is the mouse, and this is the interface. And we track these neurons to see how the mouse learned and how the brain changed as a result of this learning.
Yeah, I would like to be the mouse, but thank you very
Welcome.
much. Thank you.
Welcome.
Very interesting.
Yeah, we need to solve. You guys can go there while get the short info here. Yeah, you can do it.
Here.
I told you only for photographs.
So hi, welcome. So this is one of the experiments that we are doing in this lab in order to investigate the coding of audio processing in the mouse brain. So currently there is a awake mouse that has two electrodes in his brain, two different areas. One is the olfactory bulb, which is the first station for audio processing. The second one is the peripheral cortex, which is the first cortical station for audio processing. And we're using state-of-the-art electrode that enable us to record from hundreds of neurons simultaneously from the two areas. So you can see each row here is one electrode inside the brain, and you can see these spikes, which is spiking activity, and this is how neurons communicate with each other, transferring information. And these mice are very good mice, and actually very good in audio processing, but we actually interested in how they learn information is encoded in the brain. So for this aim, we are training them in a system that we built to discriminate
Though we need to finish, the other group is here.
very, very similar orders, and then we take these mice and record the activity. Yeah, the mouse is there, two electrodes.
Sorry.
Thank you.
רואים משהו?
כן, אבל אני שלא תעשה פלאש קודי.
לא פלאש, ככה.
The red group is coming to Mizrahi lab. He's the dean, but he's also a scientist, a great one. So this is his lab. Please come in. We will wait 1 minute for another visitor.
Yeah, no problem. So, you're welcome to come in and welcome to our lab.
Minutes. Wait 30 seconds.
שאני אלך לשם?
Okay?
שאני אלך לשם?
Yeah, that's it. look into it.
Should I go to that side? Or continue?
This is your decision. Naomi is now over there.
That's the group that was here, right?
Yes.
Right, and the
So
group that was...
this is the group that was in Naomi Chaviv, and now it's come here.
There's another group? Oh, no, there's two.
Yes, of course.
Ah, so no, okay.
Danny.
Are we waiting?
No.
Okay.
You can start. Just over there.
Which way, Ramsha?
This way. Yeah.
Okay.
Okay, wow, this is amazing. Thank you for coming. So, in my lab, generally, we study Alzheimer's disease. And we really try, as you all know, this is a very devastating disease that affects millions worldwide. And currently, we have no cure, no effective treatments. So, our goal is to find an alternative way to, alternative therapeutics. So what that actually said, it means that we had to try and study Alzheimer's in a different way. So let me actually show you here. So for what does Alzheimer's do is it very dramatically affects these special cells in the brain called nerve cells, which you can see here, they're actually dying during Alzheimer's disease, which causes the dementia and the cognitive decline and all the other devastating things. And what we said, though, is that if over 30 years people have been studying Alzheimer's, mainly focusing only on these nerve cells, we really need to think about this differently. And if you actually take a slice of brain and look at it under the microscope, you would see, so this is obviously a cartoon, but you would see that the nerve cells, they're not living in a vacuum. They're actually surrounded by many, many different other cell types. This is a our brain is a very rich tissue. We have you can see the cells in all the different colors around them. So what we are saying is we can't just look at one cell and ignore its environment. If the environment of the cell is becoming toxic and inflammatory, we wouldn't find a cure to the disease if we don't understand all of this together. So for that, we actually needed a technology that will enable us to do that. Enable us to look at every single cell type separately and profile it in a healthy brain, in a diseased brain, and understand what drives disease. So what I'll show you now is our technology that's been developed especially for this. Not only for Alzheimer's, but for profiling brain cells at the single cell level. So, and what we're doing is we're a molecular lab, we're measuring molecules called RNA, which are highly informative to what the cells are doing, what's happening in the brain. And what we are doing is that if traditionally people have been measuring RNA by taking a piece of tissue, mashing it all up, and looking at everything at once. So you can think about it as a fruit shape, where if you have a rotten fruit, you might feel its taste, but you don't know where it's coming from. So what we want to do is we want to look at every cell separately, more like a fruit salad, that we can look and taste and feel the texture of every fruit separately. And really, and back to cells, really finding the cause and the drivers of disease. Okay, so let me show you what we're using for that. It's a new technology. We're using microfluidic devices. So this is kind of hidden in our clean room, but you can stand all around and look at this. You can see it's a demo running on our computer. Maybe Danny, you want to help me? So this is Danny Kitzberg.
Hi. In old days, if we wanted to look at individual cells, we'd use something like a 96-well plate, which we had to capture 96 cells, and then we could do analysis on them. The state-of-the-art technology we're using now is using microfluidics. The bubbles that you see going to the left are oil droplets, and each oil droplet captures one specific cell, which we then label with a barcode, and then we can take all the droplets together and analyze 10,000 or 100,000 cells, and each one has got its own barcode, so we'll be able to analyze all these cells in one go and be able to see which cells are affected in a healthy, in a diseased individual compared to a healthy individual.
Yeah, so just to clarify, in our brain we have millions and millions of cells. So if we kept analyzing 96 cells at once, imagine how much work we need to do in order to profile 1,000 cells or 10,000 cells. Now we replace this with a microfluidic device that we can, in one go, look at 10,000 cells. Okay, so this has given us the power to really build highly detailed maps of the brain, which actually what is the output of this machine is we use next generation sequencing, and we generate tons of data, which you can see here, like some students kind of sitting. A lot of our work is actually computational and not experimental. We can't show you that, but we're doing big data analysis, and at the end of the day, we're building maps, cellular maps of the brain. And these cellular maps actually already showed us many great leads on the link between the mechanism of Alzheimer's disease. We have many new discoveries now. So the last part is actually taking these predictions and translating them into and testing them in a lab, and which will show this. Anna will show that at the end. But I want to stop for a second and just kind of a detour into what Masha will show us now, which is what we actually did during the COVID-19 pandemic when it started. We sat and we said, how can we actually help during this pandemic? And we said that we have actually, we're not studying viruses, we're studying the brain, but we do know a lot about this molecule called RNA. And this is the molecule that actually is profiled when we're testing if somebody is positive or negative for COVID-19. What we're actually profiling is it's the RNA of COVID-19. So here, Masha.
Okay, so during the peak of the outbreak, many labs here in Israel ran out of the reagents that are necessary to perform the diagnosis. So we came up with the idea to test the reagent we usually use in our lab to replace for the missing one. So I want to show you what we're talking about. You can see here brown suspension. Those are beads. Those beads have two properties. First, they can bind RNA. That's the molecule we need to test for coronavirus. Secondly, those beads are magnetic beads, so by applying them to a magnet, we can easily separate the beads with the bound RNA. You can see the beads at the bottom, and separate it from the unwanted molecule, so it will be available for the next step of the testing. So we show that this method can actually replace existing commercial kits. It's very efficient, it's very cheap, mostly homemade, and we tested with clinical samples to show that it's actually really accurate.
Yeah. So this turned out to be an amazing work we did with Hadassah Medical Hospital, and we kind of tapped into an existing collaboration of the Hebrew University with Hadassah, that they're doing a lot of efforts on COVID-19. And for us it was a thrilling journey in how can we really speed up our research and within a few weeks go from an idea to a product, which usually, takes years in clinical research. So that's something that's unique about, I think, times of crisis. We're really excited about this and continuing to work about new developments, but I'll actually go back to our Alzheimer's research. So I remind you, we're building maps of the brain, we're doing a lot of computational data.
Until